Let us discuss the structure and the anatomy of the vascular system, the blood vessels of the body. Now, the blood vessels of the human body are a closed circulatory system. We have arteries, which receive blood from the ventricles, and then they bring blood to the either to the lungs or to the rest of the body. And then we have veins, which will be returning blood from the lungs or from the rest of the body and bringing it back into the atria. So I think it's helpful to kind of tie veins together with atria. Remember that atria are the receivers of blood. They sort of passively allow blood to simply, enter into the heart, just receive it with very little pressure. And that is true of the veins as well. They are meant to be open and low pressure, and they are receivers of blood as blood is being returned back to the heart. Now, the structure of the veins is in keeping with this idea. They are low pressure, they have very little musculature, but one of the things we want to point out right away is actually the outermost layer of the veins, the tunica externa, is quite structured, and rightly so. This is the structure that the veins really have. So their tunica externa, the outermost layer of the veins, must be thicker. If we go over to the artery side, the tunica externa is actually going to be quite a bit thinner because they do not require as much structure. They're going to be getting a lot of their structure from the next layer in, which is a muscle layer. Now, the tunica externa, this outermost layer, is made primarily of collagen. Collagen gives strength. Collagen gives strength to anything that it's a part of, and therefore it shows the idea that a lot of the strength of a vein actually comes from having a nice thick tunica externa. Well, the tunica media is a completely opposite story. The tunica media is quite thick on the artery side, but it is quite thin on the venous side. So it turns out that a lot of the structure of an artery comes from the tunica media. Now, the tunica media is made of muscle. So this points to the completely opposite roles of the veins and the arteries. The arteries are very muscular, thick tunica media, lots of muscle. On the artery side, we decide where blood is going to go. We are going to be in charge of, the arteries are going to be in charge of blood distribution, blood pressure, a few other physiological processes that we'll be discussing. And they're muscular. So on the venous side, you don't want them to be a high pressure situation. Not a lot of muscle here. There's just, they're receiving blood, they're passing it along, and actually that's quite a challenge that they have to deal with, which we'll be discussing later on. All right, so we're going to use a third color here. In purple, we're going to be drawing the endothelium. The endothelium is going to be the same no matter where you are. So this is called the tunica interna, tunica intima, or the endothelium. Commonly, it's just called the endothelium. It's made of simple squamous epithelium, and it's meant to be a smooth, frictionless passage for blood. A smooth, frictionless passage for blood. This is the one layer that's going to be the same in all of the structures we're going to be discussing. The tunica intima, tunica interna, or endothelium is going to be just simple squamous epithelium, and it's meant to be a very smooth. It's not meant to, like, for example, catch any of the blood. It's just meant to pass it along freely. Now, the endothelium is going to be surrounding the central blood-containing space called the lumen, the central blood-containing space. And I wanted to kind of point out the diameter of the lumen and see how very much smaller that it is in the artery side versus the venous side. Okay, so let's discuss the three different types of arteries first. The first type of artery we're going to be discussing is called the conducting or elastic artery. And the most famous example of this, of course, is the aorta. Most of the arteries that are very close to the heart are going to have a significant amount of elastin fibers inside of the tunica media. So the tunica media is going to be muscular, but a significant portion of the structure of the tunica media is also going to be elastin fibers. And this, of course, gives them flexibility so that these structures, these arteries, the aorta, for example, is able to easily expand when there's a surge of blood being pumped in from the ventricles, and then it's able to snap back together in a nice, healthy way, especially when you're young. Everything loses its flexibility to a certain extent as you get older. Those elastin fibers begin to break down, kind of like an old rubber band. And therefore, especially if a person has not been exercising their heart very much, you might lose a lot of that expandability of the aorta and other conducting arteries. And so, um, yeah, you might end up with a condition called arteriosclerosis. Arteriosclerosis is the hardening of the arteries. Sometimes this can be due to calcification, which can definitely happen as you get older, especially you end up with calcium deposits that starts to harden things up. But one of the ways that arteriosclerosis can occur is through those elastin fibers kind of breaking down. Now, these are called conducting arteries. It's pretty obvious why they're called elastic arteries, but they're also called conducting arteries because they are meant to have a nice ability to simply pass blood on with very little resistance. Really, these are not going to be the arteries that are going to be constricting all the time, for example. They're much more likely to expand and then come back together into their natural condition, into their natural diameter, for example. But some things can sometimes go wrong. Sometimes because of arteriosclerosis or that hardening of the arteries, you can start to have a tear in the blood vessel wall. And this would specifically be a tear in the endothelium. So when you have a tear in the endothelium, what happens is the blood starts to pool inside a pocket. Basically, it starts to create a pocket that can actually start to expand. And this is called an aortic dissection. An aortic dissection. It can present itself as chest pain. So oftentimes people will be thinking they're having a heart attack. It's not specifically a heart attack, but it does actually have to be repaired, usually through open heart surgery, although I have come across surgeons are able to sometimes do this laparoscopically. What they actually have to do is replace a portion of the aorta, which is a pretty serious and in-depth surgery, of course. Anyway, so this is an aortic dissection, and it has to be replaced within about 24 hours in order for the person to recover. Otherwise, that pocket that is between the endothelium and the tunica media begins to expand and leads to basically the flow of blood simply not being where it's supposed to be. It kind of looks like an aneurysm, but it's not exactly an aneurysm. It would lead to eventual blockages and the inability to properly pass the blood on or conduct the blood onto the next structures. So just a reminder, the first artery we're taking a look at is the conducting or elastic artery. The second artery we're going to take a look at is called the muscular artery or the distributing artery. This is also called the named arteries. Most of the arteries that you kind of know the names of, like for example the brachial artery or the iliac artery or the renal artery, these are they're bringing blood, they're distributing blood into fairly substantially large areas of the body. So they're pretty big vessels, and they're the ones that you know the name of. And then branching off of this, you're going to have smaller arteries called arterioles. So I wanted to kind of show, then we're going to switch to red here because arteries are typically portrayed in red because they typically bring oxygenated blood to the tissues of the body. So we're going to show two different versions of this. One of them where the distributing artery is kind of narrow and the arterioles are thin. And then one where the distributing artery is actually wider and the arterioles are a little bit wider themselves. And so on the left-hand side, we are drawing a situation of vasoconstriction. And then on the right-hand side, we're drawing a situation of vasodilation. I wanted to describe, basically the practical side of why this would happen. It might help us to go all the way back to a lesson that we learned earlier with the sympathetic nervous system. Remember that fibers come from the sympathetic side of the autonomic nervous system, and they innervate these specific muscles, the tunica media, in the walls of these vessels that we're discussing right now. And when you experience a nervous impulse going through there, when there's an action potential passed on through these sympathetic fibers, this in turn causes vasoconstriction. So vasoconstriction requires action potentials. Vasodilation requires that those action potentials cease. First of all, if you're vasoconstricted or if your arteries are narrower, your blood pressure is going to be higher. And conversely, if your distributing arteries are wide and your arterioles are expanding and carrying more blood, then your blood pressure is going to be lower. Now, secondly, we're going to be talking about shunting blood or distributing blood to various parts around the body. And if you think about it, you kind of have to work your way around this one a little bit. But the idea behind a vasoconstriction means that you are not shunting blood in the direction of that area of the body. When you open up the vessels, when you vasodilate, you are bringing more blood into that particular region. So the practical example of this would be you've just eaten a big meal, you need to bring lots of blood into, for example, the walls of the stomach are very muscular and they need to churn the food and they need to be moving the food along appropriately. Therefore, those muscles are actually going to be commanding or demanding more blood. So you're going to shunt blood towards those muscles, the muscles in the walls of the digestive tract. And you might be shunting blood away from, for example, your skeletal muscles. So it would be difficult to exercise after a big meal. It would be difficult to be able to distribute blood appropriately to both your entire digestive system and your skeletal muscles for exercise. Probably the primary reason why it is not recommended to work out right after a big meal. And finally, one of the other implications of vasodilating or expanding the blood supply into a particular region of the body is inflammation. And the inflammatory response is dependent heavily on the way that blood is moving around. The more blood you have coming into a region, the more ability that tissue has to become inflamed. So we're going to be discussing this more down the line. But inflammation basically is redness, heat, and swelling. And all three of these things, redness, heat, and swelling, are going to be dependent on blood vessels. The swelling of an area of the body is because of fluids being brought into the area by the blood supply. So you expand the distributing arteries, you just expand the venules, the arterioles bringing the blood into the area, and that brings more fluid. And at the same time, if you have lots and lots of arterioles that are expanding, that causes the tissue to become more red. So that's where the redness comes from. Right, so let's move on to capillaries. There's two big topics that we discuss when it comes to capillaries. First of all, we're going to be discussing the capillary beds, and then we're going to discuss the three different types of capillaries that will be found, in those capillary beds. So in order to draw some capillary beds, we have to draw some arterioles and some venules. These would be considered pre-capillary arterioles and post-capillary venules. And between them, the first thing that we're going to be drawing is actually a vascular shunt. And the word shunt means bringing something to. So a shunt simply brings blood across from the arterial side to the venule side. And as you'll notice, we draw our arterioles in red because they typically carry oxygenated blood, and we carry our venules in blue because they typically carry a blood that is oxygen deprived or has a lower level of oxygen. From there, then, in one of these pictures, we are going to draw some capillaries. And you'll notice, of course, that they transform from red to blue. So what happens is these little structures here, these true capillaries, are going to be giving off oxygen and picking up carbon dioxide. That is why they turn from red to blue, which is actually how the blood sort of looks. When blood is fully oxygenated, it's bright red, and when it's deprived of oxygen, it kind of takes on a darker purplish hue. All right, so what we're drawing here then, all these little networking vessels that we're drawing are going to be our true capillaries. And again, that tube going straight across from one side to the other is the vascular shunt. Typically, the vascular shunt, there's usually only one vascular shunt, but there's between 10 and 100 true capillaries per capillary bed. Now, the true capillaries are where the actual exchange is going to be taking place. The other capillary bed we're going to be drawing up above here, we're going to draw all of those true capillaries actually in black, indicating that there actually is no blood moving through them in this case. So the blood supply is moving straight from arterial to venule through the vascular shunt, but is not actually moving through the true capillaries. So therefore, there is no actual exchange taking place in here because the true capillaries are the primary place where you're actually going to be exchanging, for example, oxygen into the tissues and then carbon dioxide back out of those tissues. Now, lastly, at the roots of each of those arterioles, we're going to go ahead and draw our precapillary sphincter. Our precapillary sphincter. Now, a sphincter is a general term that means a muscle that surrounds a tube. So in this case, it's a muscle that surrounds the base of the tube and basically controls whether blood enters into that tube or not. Now, up above, those precapillary sphincters are going to be constricted. They're going to be contracted. They're going to be closing off those tubes so that blood does not enter in. In the capillary bed down below, where you actually see blood flowing through those true capillaries, in those cases, the precapillary sphincters are actually relaxed and open and allowing blood through. So one helpful thing would be to make a comparison between A and B. A up above, blood is not going through the true capillaries. B down below, blood is making its way through the true capillaries, and therefore what we can say is that in B, the surrounding tissues are actually being fed. They're actually getting their oxygen, getting nutrients. They're able to give up their carbon dioxide and their waste products. In A, there's very little feeding of the tissues going on. Therefore, these tissues are going to be deprived of oxygen. They're not going to be able to get oxygen or nutrients or give up their carbon dioxide or give up their waste products and so on. So it's possible that these tissues are simply sort of quiescent. They're not metabolizing actively at the moment, and therefore they actually don't require any kind of a blood supply in that moment. There are three different types of capillaries, and keep in mind that when we look at true capillaries up above, any of these three types of capillaries could be part of that. And so therefore, if we're asking for types of capillaries, there are three. Now, all of the capillaries are made of pretty much one single layer of cells. Sometimes there are pericytes or little muscle cells to kind of hold everything steady. But for the most part, the composition of capillaries is simply simple squamous epithelium, although the way that the cells actually look and appear and interact with each other can differ depending on the type of capillary. The first one we're going to be drawing are continuous capillaries. And we draw in purple again those simple squamous epithelial cells. We're going to be drawing a similar set of 6 cells, kind of overlapping each other as you see, with these tight junctions. But in between, sometimes there's a little bit of a crack in there. These are called intercellular clefts, and we're going to be drawing those in black. So this is a continuous capillary where most of the cells are overlapping with tight junctions, but you have some intercellular clefts. The intercellular clefts are helpful because they're able to easily pass water and fluids and salts and acids and things like that back and forth into the blood supply or back out of it. However, those intercellular clefts are actually really helpful because they're wide enough for those white blood cells to squeeze through. Remember diapedesis. And so inside of a blood vessel, of course, you have blood, but just outside you will have interstitial fluid. And typically infections and pathogens living in your body oftentimes aren't living in the blood, but rather in the interstitial fluid. So white blood cells, remember, need to be able to quickly and easily move in and out of the blood supply in order to migrate their way through those intercellular clefts and find infections and pathogens and things like that. And so continuous capillaries are probably the most common type of capillary found in the body. They're going to be found, for example, buried in the skin, buried in the muscles of the body. They are the cells that actually make up, these are the capillaries that make up the blood-brain barrier. So these are the capillaries that are typically found adjacent to neurons in the brain. However, in the brain, you're not going to actually have those intercellular clefts because the neural tissue is quite sensitive to things like infections, and therefore you cannot risk having a free and open blood supply and things being able to migrate in and out of the brain. And therefore the brain, there are no intercellular clefts, there's just those tight junctions, and in those cases the cells are quite smooth. So the blood-brain barrier is made half of continuous capillaries that are kind of specialized for that purpose, and then the half of that layer would be the astrocytes that are serving the neurons in the brain. The second type of capillary we're going to be drawing is going to be a fenestrated capillary. We're going to replicate as best we can, not being an artist. We're going to replicate the first set of capillaries, and we're going to replicate the intercellular clefts as well. And so we're indicating that yes, white blood cells are still able to squeeze in and out of our cells, of our blood vessels here at these capillaries. But we're also going to draw some hole punches, and we're going to draw these in purple. We're going to draw hole punches that move right through the cells themselves. These are called fenestrae or fenestrations. They're about 70 nanometers in diameter, so not large enough for a big old white blood cell to squeeze through, but they are large enough for really fast filtration of things like salts and acids. So We'll typically find fenestrated capillaries in places like the kidney tubules, and we will be discussing those more down the line. The last type of capillary we have is the sinusoid capillary. The sinusoid is going to have big old Swiss cheesy holes everywhere. The intercellular clefts are absolutely massive. So again, we're going to be replicating those 6 cells that we've been drawing, and we're going to do our best to draw just a few of our tight junctions. And so there is a little bit of an overlap between these cells, but the cells are going to be narrower. The gaps between them, the intercellular clefts, are going to be huge. And there are also going to be fenestrations, but these fenestrations are going to be much larger than they were drawn before. So big old intercellular clefts, very large holes, tunnels going straight from one side to the other. They are still called fenestrations, but these fenestrations are much, much larger. So sinusoid capillaries are going to be found in places such as the liver, or I think the place where we're most familiar with them is going to be in the red bone marrow, where the elements of the cells, the formed elements of the cells, are going to be created within the red bone marrow, and those formed elements need to be able to quickly move into the blood supply, and therefore you have to have a pretty Swiss cheesy endothelium there. You have to have pretty Swiss cheesy cells in order to get those elements into the blood as quickly as possible. So sinusoid capillaries. All right, so let's go ahead and finish up with the very last structures we're going to be drawing, which are the veins. Now, the first and largest of the veins are the vena cavae, the very large tubes that are just adjacent to the heart and deliver blood directly into the right atrium. And then the third, like the smallest of the veins, are going to be the venules, which we've already been drawing here. Well, let's take a moment to draw our named veins. Our named veins is just kind of like a type of vein that you typically think of when you think of a vein. And again, going back to our muscular arteries, these are also called named arteries. So the named veins are going to be typically adjacent to arteries that also bring blood into large swaths of the body. The named veins are going to be draining blood from large swaths of the body. So typically, we think of the veins being in the legs. The legs have a significant and unique challenge because they have to bring blood back in not muscular conditions, all the way from the tips of the toes all the way up the legs against gravity. So it's a long structure. It's against gravity, especially if you stand a lot for example, for your job. And therefore the challenge is unique. So it's not muscular, it's quite long, and it is going against gravity, especially if you're talking about the legs. So typically when we talk about the named veins, we are going right to the 2 largest veins that we usually find in the thigh, which would be the femoral veins and the saphenous veins. Now, the femoral vein is considered a deep vein. It actually, just like the name suggests, it's deep. It's buried within the muscles of the legs. But the saphenous vein is pretty superficial and medial and is actually relatively close to the surface of the body. So the femoral vein, a deep vein, the saphenous vein, a superficial vein. Now, just to kind of clarify the difference between the two, about 90% of the blood that is returned up the legs is going to be going through the femoral veins. The saphenous veins only return about 10% of the blood up the legs, and therefore they are not as significant or as important, but they both have their challenges. Both deep veins and superficial veins have their challenges. And the challenges kind of surround what we're drawing here, which are veins, which we typically draw in blue. And we're going to go ahead and draw venous valves. We're going to draw a venous valve that is closed and a venous valve that is open. As long as the venous valve is open, blood can flow through, but once the venous valve is closed, blood begins to kind of bounce back into it. The purpose of the valves, as is always true of all valves in the body, is to prevent backflow. And so this keeps the blood moving upward in the direction of the heart. And secondly, the second thing that we're drawing in this picture is a skeletal muscle. Now the skeletal muscles are involved in milking the blood and keeping it moving along. There is no directionality that is chosen by the skeletal muscles. They are simply milking it. So, as long as you are moving the muscles of your leg, you will be continuing to move the blood along through here. It's one of the reasons why we are concerned about people who are bedridden or people who are wheelchair bound. We are concerned about the blood flow through those legs specifically. It is difficult when you're not using your muscles quite a lot to continue to move the blood along. So, again, the two physiological functions that we're looking at here is the milking of the skeletal muscles, and this would be especially significant with the deep veins, which do tend to run directly through those muscles. And then the venous valves. The venous valves are going to be very significant, especially for those superficial veins, because those are going to be the veins that are not going to have as much of an opportunity to be milked by the skeletal muscles. Therefore, one of the challenges of the saphenous vein and other superficial veins is they tend to form they tend to kind of bulge outward in places where the valves have started to kind of fail. Those types of superficial veins are so dependent on their valves that the valves kind of start to break down. They are fairly simple, just basically an invagination of the endothelium, and therefore under pressure situations, sometimes they start to fail, and therefore the vessel starts to kind of dilate. And this bulging outward, which has a specific look to it, is called a varicose vein. You will see those on the surface of the skin because they always happen to superficial veins. Now, the deep veins also have their issues. They tend to sometimes accumulate things like a small coagulation event, especially in folks who have issues with expressing too much coagulation in their blood. These tend to gather in places where the blood flows a little bit slower, such as the legs, or where there's more of a challenge, such as the legs, and therefore your blood tends to clot in these areas, especially close to places like venous valves. You might actually form quite a significant and large and long clot, part of which might break off, make its way up through the valves straight into the inferior vena cava and directly into the heart, which then makes its way around into the lungs and could form a pulmonary embolism. So a deep vein thrombosis is therefore tied to an issue in the lungs, a pulmonary embolism, because the clots that have formed in the legs could eventually make their way into the lungs. It's something that we're actually quite concerned about a lot when somebody has a clot in their lungs, a clot in their legs. Deep vein thrombosis. So a primary concern of the saphenous vein would be things like varicose veins, but a primary concern of things like a deep vein would be a deep vein thrombosis, or a DVT. So the unique challenges on the venous side are that you have to return blood, and there's lots of challenges to that, especially when you're talking about certain structures of the body. But on the arterial side, just to remind you, it's actually quite muscular. There's a very different set of challenges, and one of the things we want to consider is, where does the control of blood flow actually take place? So oftentimes when I would ask students this question and kind of consider on it a little bit, oftentimes they would come up with, well, the precapillary sphincters. The most local control of blood flow is going to be those precapillary sphincters, where you have an extremely tight decision of deciding, well, okay, this blood is actually in this capillary bed, but we're going to decide whether the local tissues receive it or not. Close up those precapillary sphincters, no blood supply. Open up or relax the precapillary sphincters, and yes, blood is sent to that tissue. Now, the other two places where you can significantly control the flow of blood, one of them would be in the arterioles. Although they are tiny, they still have a significant amount of tunica media, or muscle, in their walls, and therefore they are capable of vasoconstriction and vasodilation. And of course, those distributing arteries themselves are capable of either closing off large swaths of the body, large regions of the body, vasoconstricting and not sending blood there, or shunting blood in the direction of other regions of the body that happen to require it in that moment. So again, the three places where we control blood flow would be the distributing arteries, or the muscular arteries, the arterioles, and those precapillary sphincters.